Hello everyone, and good morning or good afternoon, depending where you are. And welcome to our fourth webinar about the different health benefits of marine ingredients under the project Marine for Health. So after health brain, gut microbiota, and stress management, I'm really excited about our topic today. So the topic is about the metabolic syndrome, and we will have these three amazing speakers today to present their different studies. So my name is Rachel Durand, and I am the project manager of Marine for Health for the cluster NCE Blue Legacy. We are a national cluster located on the west coast of Norway at Ålesund, and our main purpose is to increase and improve the use of marine resources, mostly for a high-value ingredient for human consumption. So now we are currently working with more than 50 different members from industrial but also academical, from all the value chain, so from the fishermen to the ingredient producers. The Marine for Health project is focusing to establish and increase the scientific expertise in health effect of different marine resources. And that's why we are here today. So before to start our webinar, I just want to remind you that the webinar is recorded and you will have access to the video, but also to all the presentations from our speaker on our website, legacy.no. I also want to tell you that you can use the Q&A box, but also the chat function to ask all the questions that you want to our speakers, and I'm sure they will be happy to answer it during our discussion session at the end of the webinar. So now we are going to start the webinar, and we are going to start with our Canadian friends, Jack Gagnon and Yan Wen Wang. So the Dr. Yan Wen Wang joined the National Research Council of Canada as a research officer in 2004. He has been an adjunct professor at the University of Prince Edward Island and Dalhousie University in Canada. His research interests have been in the areas of metabolic health, natural health products, and functional ingredients, marine and plant-based protein product development, and quality assessment. He also received numerous research project funds by federal and provincial funding agencies and private sectors. He received several awards from the National Research Council of Canada and has published over 70 peer-reviewed research papers and 4 book chapters and provides over 100 presentations. So I'm really happy to have him today. He will also be joined by the Dr. Jack Gagnon. He's a senior researcher at Valorise at Shipyard in Canada for more than 15 years, and he acts as the scientific director of fishery and marine co-product divisions. He has now more than 40 years experience in the international research, and he was awarded by the New Brunswick Innovation Funds for the excellence of his work. His research interest is in the marine ingredient for human health, in diabetes, and also in obesity. He is collaborating with the Dr. Yan Wen Wang from more than 10 years, and his career in New Brunswick is also held an adjunct professor position at the University of Moncton. So now, Yan Wen Wang and Jacques Gagnon, the floor is yours.
So thank you, Rachel, for your nice introduction. And I'll start first, and then Jack can follow up and add more information. And as you said, today we are talking actually a part of our other collaborative research in 10 years, or more than 10 years, And this is just a part of that. And we talk about the milk hydrolysate for obesity and diabetes. So metabolic syndrome is actually complex. It's a cluster of different metabolic disorders, mainly include obesity, diabetes, and dyslipidemia and hypertension. So today we are mainly focused on obesity and diabetes. And we know that over the years, in the last several decades, obesity and diabetes has been increasing very fast. And for example, I think last 20 years, start from 2000 to 2019, they over 1 billion adults are overweight, and of which at least 300 million actually are obese. And more seriously, this situation has been quickly getting to children and adolescents. And because of obesity and diabetes cause of death increased by 70% globally in the last 20 years. So this is I said, there's two different diseases contributed to metabolic syndrome. And all those actually also contribute to heart disease, so which actually increased by almost 30% in the last 20 years. So certainly it's a serious problem. So our research, this whole project is actually led by Dr. Gagnon. And we are Valorise source biomass and bioproducts derived from marine species. And we start from collection, processing, fraction, extraction, and chemical preparation. And also, more importantly, we do different in vitro assays to screen what kind of potential applications or have benefit. And based on that, we can develop a product, make a value-added products from, more importantly, from marine-based bioproduct. And one of them is glucose-stimulated insulin release or secretion in pancreatic beta cells. So in this screening, we did several times. Talk about first three screenings, and we use red beta cell lines as a in vitro assay model. And of course, we first we test the toxicity, and then once we know non-toxic range, and within that range, we test the effect on glucose stimulated insulin secretion or GSIS. Using ELISA kit. So we tested at least 82 different extracts and fractions. In the first screening, we found actually 32 fractions or extracts. They increase insulin secretion by at least 20%. Others are either toxic or either not so effective. So we in the second screening, we needed to test these 32 lead candidates. I use a little bit different condition adjusted based on first screening. And this time, we select top 13 lead candidates. That's increase insulin secretion at least by over 40%. So the list here is actually a product cost at the 13 extracts or fractions. So 2-1 P plus A, that is a hair milk protein hydrolysate. So I labeled out so we can. And then we continue to do third screening and further confirm the effect on GSIS. And this time we also add shrimp oil. So we select the top 9, and again, hair milk hydrolysate show quite a positive effect as on the list of these 9 good candidates. So The hydromelt protein hydrolysate actually prepared from weight of fresh hydromelt. I put it in a bioreactor with alkalos, and then after reaction, simply dry up. That's my understanding. And after that, do the chemical analysis. We found it contain very high content of protein, like 71% of protein, and 11% of lipids, and enriched with EPA and DHA omega-3 fatty acids. And also very important to note that in amino acids, it's really highly enriched with arginine. The arginine has lots of benefit, also including on weight change and glucose control. So based on that, we make a different diet, and we have a high-fat diet to induce obesity and insulin resistance, and add a different concentration of hydromelt protein hydrolysate, such as 15%, 35%, and 70% replacement for dietary casein. So we call this diet H15, HPH35, and HPH70. So we adjusted fat, starch, protein, and energy in the diet. So that all the treatment diets contains similar or same content of protein, fat, and carbohydrate, and also energy density compared to high-fat diet. And we use a low-fat diet as a normal control to verify our high-fat diet-induced model is successful. So we know that diabetes, especially type 2 diabetes, that is, when this happens, they have to change many parameters or metabolic phenotypes. And listed later of this slide, for example, glucose intolerance, and increased blood glucose, insulin, leptin, and free fatty acid, triglyceride, and cholesterol, and decrease blood adiponectin. Of course, they have more than those, and a bunch of other biomarkers are changed by insulin resistance or diabetes. But in our studies, we focus on those metabolic biomarkers to evaluate how our products can improve or change insulin resistance in animal model. So we use the C57 black mice. Those mice actually give a high-fat diet at age of 5 weeks. And after 11 weeks, they are basically developed insulin resistance. And we measure body weight and glucose, blood glucose. Based on those two parameters, we divide animals randomly into 4 groups, high-fat control, HFC, and 3 groups of treatment, where that, as I said, the diet casein replaced by hydromelt protein hydrolysis, HPH15, 35, and 70. And we one group of animals, they given low-fat diet all the time and used as a normal control. So we give animals 10 weeks treatment and measure food intake, body weight, semi-fasting glucose, and oral glucose tolerance. And by the end, we fasting animals overnight and measure fasting blood glucose, insulin, leptin, adiponectin, free fatty acids, and lipids. So this first we look at the body weight change. So you see from this figure that this blue line is a low-fat control. But all the time, from beginning to the end, the body weight was significantly lower than high-fat control is a red line. And two treatment, HPH15 and 35, they tend to be lower, especially 35, but it's not significant. However, the HPH70 get a lower body weight after 2 weeks, and the effect remains all the time until the end of the study. So in this group, basically over the treatment time, they didn't really increase the body weight. So by this time, when you think that the weight reduction is due to a version test of a diet, or they have some side effect to the animal didn't eat enough, or other reasons. So the next slide showed that the food intake. So here, that the again, the low-fat control, they tend to eat more all the time. That's predictable. And compared to high-fat control, the red line, and this green line is HGH 70, that actually tend to eat more rather than less all the time. However, this group had lower weight gain or body weight. And the other two groups didn't show a significant effect compared to high fat. They have a very similar food intake and also a similar body weight. So we measure the semi-fasting blood glucose. It's for a 4 to 6 hour fasting. And after that, we measure blood glucose. And again, it's all the time, it's a blue line. The low-fat control shows significantly lower semi-fasting blood glucose compared to high-fat control, it's a red line. And the other two groups like HGH 15 and HGH 35 showed some effect and a significant difference at a few time point. But more importantly, HGH 70 showed a significantly lower glucose after 2 weeks, and again 5 weeks, and all the time to the end. So actually, look at after 5 weeks of treatment, this group showed quite similar blood glucose compared to normal control. And we measured oral glucose tolerance twice, actually, 5 weeks and 8 weeks, and similar effect. But here I just show one of them is 8 weeks treatment. And we give 2 grams per kilogram body weight of glucose, and then measure blood glucose at 30 minutes, 60, 90, and 120 minutes to see how oral glucose can increase blood glucose concentration. But if you have better insulin sensitivity, and you can control glucose better. So you see, over the time, low-fat control or normal control, they had a significant lower blood glucose compared to high-fat control all the time. And the treatment tend to decrease blood glucose, especially HGH 70, but statistically they're not significant due to variations. And however, when we calculate area on the curve of oral glucose tolerance, and then we see significantly higher in high-fat control compared to low-fat control. And treatment HGH 70 significant decrease compared to high-fat control. So I just emphasize that we did the statistical analysis actually compare low-fat and high-fat control using t-test, and then compare those 4 groups use one way ANOVA and multiple comparisons. So I labeled here is differently. So we see this label means that's a difference between those 2 control groups, and this later that compares 3 treatment groups to the high-fat control. So actually also fasting blood insulin was several-fold higher, pretty like 3-fold higher compared to normal control. And again, HGH 70 decreased blood insulin compared to high-fat control. And other 2 groups didn't show a significant difference, although HGH 35 also tend to be lower. And a similar pattern for fasting blood leptin. And fasting blood glucose significantly high in high-fat control compared to low-fat control. And then treatment HGH 70 significantly decreased compared to high-fat control, and no effect or significant effect in these 2 groups. And the blood cholesterol was significantly high in high fat control compared to low fat. And then again, treatment of HPL70 significantly decreased compared to high fat control. And other two groups didn't show a significant effect. And we also calculated the hormone insulin resistance index and hormone beta function index. These two indexes are commonly used to further evaluate insulin resistance and beta cell function. Again, see here is a high fat control that are several-fold higher compared to a low fat control or insulin resistance index. And the treatment HP35 tend to be lower, but due to variations, not significant. But HPL70 significantly lower than the high fat control, again closer to normal control. And the beta cell function is even more dramatic. So its beta cell function was decreased dramatically in high fat control compared to normal control. And then you have a treatment HPL70 restored beta cell function at significantly better than high fat control. And other two treatment groups tend to be better, but didn't show a significance statistically. So we also measured serum adiponectin, and which was lower, significantly lower than high fat control, that as predicted, the high fat control or obesity or diabetes, they tend to decrease adiponectin. However, our treatment didn't show a significant effect, and the free fat acid didn't show effect between two controls, and no treatment effect either. So quickly summarize that the take-home message is from this study is that helminth hydrolysis improves insulin resistance and the glucose intolerance in diet-induced obese mice. The effect might be a result of lowered weight gain or improve the beta cell function and peripheral tissue insulin sensitivity, or could be a combination of all those together. So at this moment, we don't know it until we have further studies. So also we need more studies to identify the bioactive components, which is responsible for the observed beneficial effect. For example, protein, peptides, amino acids, fat acid, and antioxidants, and other things that could be further identified. And also what the mechanisms that are responsible for this benefit. So we have here thank of my colleagues and the in the team. That's this is a part of our team members, and Dr. Samia, Momita, Mr. Shelley, and Dominic, and Mr. Ludovic, and Mr. Claudia, and Dr. Johnson, Steve, and Vanya. All those directly or indirectly contribute to this project. And we have funding support from Atlantic Canada Opportunities Agency. That's the main funding for this whole project, bigger project. And Valorise Research Institute and the National Research Council of Canada for the support. And the picture shows a part of a team. I'm pretty sure we missed some people, but at least most people are here for this whole project. But what I present today is just a small piece of the whole project. Jack can talk more. Okay, thank you for your attention at the time, and then we address all your questions. Jack, maybe you want to say something.
So what I would say is that I will try to answer the questions. First of all, I'd like to hear the presentation of Carolin, and from there we will discuss.
Yes, thank you so much, Yanwen, for your presentation. It was really interesting. And we have some questions, but yeah, we will answer it at the end of the webinar during our discussion session. But please use the chat and the Q&A box if you want to ask some questions. So now we will go back in Norway, and we will have the presentation from Carolin Jensen. So Carolin Jensen is a clinical nutritionist with a bachelor degrees in human nutrition and a master degree in clinical nutrition from the University of Bergen. She just holds a PhD in clinical nutrition from the same university on the potential effect of a cod protein on components of the metabolic syndrome. Our research has focused on how we can use residual raw materials from the fishing industry as supplement for human use. She's now working as a researcher at Auckland University Hospital in Bergen and a clinical nutritionist at a private rehabilitation center in Bergen. So now, Carolin, if you want, you can start your presentation.
So thank you for the introduction and thank you for the invite to talk on today's webinar. So the title of my presentation is Can a hydrolysate made from cod protein improve components of the metabolic syndrome? So I will present some of the results from my PhD project where I investigated the effect of supplementation with the cod protein hydrolysate in 3 intervention studies in humans. But before I start with the studies and results, I will give a brief introduction to the background of the project. So the metabolic syndrome, as we already have been told, is a clustering of metabolic disturbances that increases the risk of diseases like type 2 diabetes and cardiovascular disease. And as a result of the global development of obesity, the metabolic syndrome has become more and more common. So there is a number of different definitions and diagnostic criteria that have been proposed for the metabolic syndrome, but most of them include increased fasting levels of glucose in the blood. You have increased blood pressure, abdominal obesity measured by increased waist circumference, as well as alteration in lipid levels seen as increased levels of triglycerides and reduced levels of HDL cholesterol. And several studies have observed that the intake of fish is associated with reduced risk of metabolic syndrome. And fish and seafood have been and still are important parts of the Norwegian diet, and we have long traditions of eating fish here in Norway. It's a good source of protein. You have the marine omega-3 fatty acids and also micronutrients such as vitamin D, B12, selenium, and iodine. And to a very large extent, the observed beneficial health effects of eating fish has been attributed to the marine omega-3 fatty acids, which we especially find in fatty fish such as salmon. However, there are recent studies that also have suggested that the protein content of fish may contribute to the beneficial health effects of eating fish. And several clinical intervention studies in humans have observed that eating lean fish, such as cod, can have favorable effects on glucose metabolism. And they've seen improved levels of fasting glucose and improved insulin sensitivity. But protein is in general, we need more protein, and the fishing industries around the world generates enormous amounts of protein containing residual raw materials. And in Norway, around 965,000 tons of residual raw material was generated within the Norwegian fishing industry and aquaculture in 2019. And these materials contain protein of high quality, and it's of interest to investigate if more of these materials can be used for human consumption. And a very promising way to utilize these residual materials is through the production of fish protein hydrolysates. But there are very few studies in humans that have actually investigated this. So in my PhD project, we have investigated the effect of low doses of a protein hydrolysate made from residual raw material from cod. And we wanted to investigate the effect on glucose metabolism in healthy adults and on the components of the metabolic syndrome in adults with overweight and or obesity. So 3 clinical intervention studies were conducted as part of the project, and I will present results from 2 of these studies today. The project was funded by the Norwegian Research Council, but it was initiated by a fishing company located in Ålesund called Firmenich Bjørge Biomarin. And they also produced the hydrolysate we used in our project. So this was made by enzymatic hydrolysis of Atlantic cod after filleting. And in all 3 studies, it was given to the participant as a powder to be mixed with water, so not as tablets. And we also gave the participants in the studies the cod protein hydrolysate in very low doses of less than 5 grams, which is based on the hypothesis that the supplement contained bioactive peptides affecting certain metabolic signaling pathways, such as pathways that lead to increased uptake of glucose into the cell. So the first study we did, we called the blood glucose study. And here we wanted to investigate the effect of a low dose with CPH, the cod protein hydrolysate, on glucose metabolism in healthy adults between the age of 40 and 65 years. And this was a randomized double-blinded crossover study, and that included 2 study visits. The intervention was a drink containing 20 milligram per kilo body weight of the CPH or a controlled drink. And the controlled drink was contained the same amount of casein, but this was given as a whole protein, so not as a hydrolysate. And the dose of 20 milligrams per kilo body weight equals to 1.5 grams of protein in an individual that weighs 70 kilos. So it's a very low dose. And the primary outcome in this study was change in postprandial levels of glucose, insulin, and glucagon-like peptide 1, or GLP-1, which is a hormone that is released into the bloodstream after a meal and increases the secretion of insulin. So in the blood glucose study, we included 41 healthy participants, 26 women and 15 men, and they had a mean age of 51 years and a mean BMI of 25. And participants indicating interest through an online recruitment form, they received information about the study by telephone, and then we did an initial pre-screening. And this was followed by a physical screening visit, which included a review of medical history. We took blood samples and we did anthropometric measurements. And then the included participants came to the research facility for a first study visit. Then we took a set of fasting blood samples, which was followed by a drink with the test or control material, and then a standardized breakfast meal. And this was followed by blood sampling every 20 minutes for 3 hours to measure the postprandial response in glucose metabolism. Then we had a washout period of 4 to 7 days before the participants came back to the final study visit, which was identical to the first visit. So I will present the main results from this study. So this graph shows the development in glucose levels from fasting until 3 hours after consuming the standardized breakfast, which included the test drink with CPH or the control. So on the x-axis, we have time in minutes, and on the y-axis, we have the levels of glucose in serum. So we did not observe any statistically significant difference between CPH and control for glucose, which is shown here. And we didn't see any difference for GLP-1, but we did find a statistically significant lower concentration of insulin in serum after supplementation with CPH compared to the control drink. However, as you might see, the difference is very small. And the clinical relevance of this finding is rather uncertain. The second study I will present is the study we call the Metabolic Syndrome study. So The overall aim in this study was to investigate the effect of a CPH supplement given daily for 8 weeks on components of the metabolic syndrome, and we had a special focus on the glucose metabolism. This was also a double-blinded randomized intervention study, but it had a parallel group design. And the participant received a daily supplement containing 4 grams of CPH or a placebo product, which was maltodextrin, for 8 weeks. Both test material and the placebo was a powder to be mixed with water, and the participants were instructed to drink this every morning for 8 weeks. And in this study, as the previous, we had a telephone screening followed by a physical screening visit, and then the included participants met on 2 study days. So 1 before the intervention started and 1 study visit after 8 weeks. And the primary outcome was fasting and postprandial glucose levels, but we were also interested in change in other components of the metabolic syndrome, such as waist circumference, fasting levels of triglycerides, and HDL cholesterol. And we also looked at insulin as well as some appetite hormones and inflammatory markers. So this figure shows the inclusion process. So 159 was interested in participating, but after a pre-screening, only 58 were invited to a physical screening visit. And of these, there were only 35 that fulfilled the criteria for the metabolic syndrome. And the inclusion criteria we had was a BMI between 27 and 35, age between 40 and 70 years, and the metabolic syndrome. So, yeah, sorry. There. So a total of 30 participants completed the study, so 15 participants in each group. Both groups had an average age around 53 years and a mean BMI around 32, 33. And these 2 graphs show the postprandial levels of glucose and insulin after 8 weeks supplementation with CPH or placebo. So we didn't find any statistically significant differences in either fasting or the postprandial glucose or insulin levels after 8 weeks with the daily CPH supplement compared to placebo. And we did not see any effect on the GLP-1 or the appetite hormone ghrelin. When it comes to the lipids, we did observe a significant reduction from baseline to end of study in the levels of triglycerides within the group that received the CPH supplement, but no difference were observed when we compared the change from baseline to end of study in the 2 groups after 8 weeks. And there were no effect on the HDL cholesterol or the other lipid markers. We did also see an increased level of adiponectin within the CPH group, but there were no difference when comparing the change in the CPH group to the placebo group. And we did not see any effect on the inflammatory markers or leptin. And if you're interested to read more, we published 2 articles last year on this study. So 1 study report on the primary outcome with the glucose metabolism, and the second one report on secondary outcomes. So to conclude on the take-home message, so if a single dose of CPH before a meal reduced the postprandial insulin concentration, but it did not affect glucose or GLP-1 levels. But as I mentioned earlier, the clinical relevance of the reduced insulin levels are uncertain, and they should be further explored. And finally, we observed that a daily intake of 4 grams of CPH for 8 weeks did not affect the components of the metabolic syndrome in overweight and obese adults. And I didn't do this project alone, so I have to say thank you to my main and co-supervisors and all the co-authors and collaborators. So thank you.
Thank you, Carolin, for your presentation. Really interesting. Also, studies a lot of work, I can imagine. So we will start. We have some questions actually in the chat. So the first one, I think it's for Yan Wen or Jacques Gagnon from Laurent Bazinet. So why choosing alkalos as enzyme, since it is a non-specific enzyme and the repeatability could be different from one batch to another?
Actually, we use a combination of enzymes. It was Protamex and alkalates.
But and it was industrial also enzyme, like
yeah, it was industrial. It was coming from we tried different sources, but we did that because we have many tissues. We have herring, we had crabs, we had shrimp. So we use this mixture for the hydrolysis. So it was not alkalis only. It was misleading. I'm sorry about this. It's Protamex and alkalis.
Okay.
Dosage itself has been published, so I can give him the article for that.
Yeah. Actually, following this question, I also have a question about the molecular weight of the peptide after your hydrolysis. It was like big peptide or really small one?
Well, we did not really analyze that. Obviously, I mean, actually, when we will discuss further, we try to, I would say, use an alternative form by using the herring milk powder. So we actually didn't hydrolyze, we used the powder, and we find a way to dry it up and to keep all the materials in the because we think in the herring milk there is much more than only proteins. There are the DNA, there are many other good things. So we think that the herring milk itself is a good product, the organ. So we try to work with the organ, and we have published that it is as efficient as if we use the hydrolysis.
Okay, so the product itself is efficient. It's more efficient than the hydrolyzed?
It's as well.
Okay.
Again, we have a published data in marine labs, and we can share that with the participants.
Yeah, actually, we have a question that have you published your findings on the in vitro effect?
Yes, we have. So we will share that to everyone. I can actually, I send you this paper, Rachel.
Yeah, I will put, if you want, I will put the link under the presentation on the website of Blue Legacy.
Yeah, that would be greatly appreciated. Thank you so much for that.
And we have a question for Carolin. To Carolin, what did you use at such low levels of CPH?
Yeah, so the first study, the blood glucose study, we had 20 milligrams per kilo body weight. So it's a very low dose, but it's based on some previous unpublished studies that someone in my research group have looked at. So they've seen effect on glucose, fasting glucose and postprandial glucose levels at these low doses. So we wanted to do a proper research project using those low doses. In the last study, so we also did a study in older adults where we tried out 4 different doses, where the highest were 40 milligrams per kilo body weight. So based on that study, we chose to use 4 grams in my last paper, so the metabolic syndrome study. And it's still a very low dose. So for future research, I think we need to use higher doses to have an effect in humans. Yeah.
Actually, 4 grams, yes, it's a low dose, but it depends of how you take it. And in your case, you mix it with water, so it was like a powder. And actually, I have a question about the flavor, because fish product taste and smell fish. So did you receive any feedback from the participant about the taste and the flavor?
And actually not.
Okay.
Some people said it tastes like fish, but we said both the placebo and the CPH smelled and tasted the fish because it was produced in a fishing factory. But you have the bitterness of the hydrolysate. I've tasted both, and you can't actually. It was okay because they added some flavor, lemon, and some sweet.
So they cannot see the difference between the placebo and the CP, right?
No, they couldn't.
They couldn't tell.
Okay.
Yeah.
Yeah, if you add flavor, I think it's better to take. But actually, it is. Do you think it will be more efficient if you take it like a capsule or tablets than to mix it in water?
The problem. So we weren't involved in the development of the hydrolysate, but what we've been told is that they had problems using tablets because you have to take so many tablets a day, and that people would drop out of the study, and the fish smell also get worse if you're going to put it into a tablet. So I think we need to develop some
new product.
Yeah, my field.
Is it like a functional food for you? You could add that to your smoothie, right?
Yeah. And add it to like oral nutritional supplements you give to elderly if you want to, but the research is very new, so especially in humans.
Yeah. And actually, speaking about dosage, in the counterwise, in your studies, Yan Wen and Jack, you use, we can see that it's only the high concentration of herring milk hydrolysate that they have a really nice effect. So do you think it can be something that we can use every day if we have to take it at really high dosage, and how we can improve maybe the efficiency of this kind of product?
Yeah, man.
Thank you. Go first. Jack, you
go. Go ahead.
Okay. It is a high dose. And the middle one, 35, shows some effect, but the most parameters didn't show significance. But also you can see the trend. But we pick up our highs, 70% of replacement of that casein is pretty high. So in terms of application, it certainly is in humans, it's a challenge based on current form. That will require quite a bit, and only if you are incorporating in diet, or you take like whey protein for fitness, right? You take spoons and you put in drink, and right, but certainly not suitable for using as a dietary supplements. For example, you take a few capsules or a tablet per day. So that's one of challenge, and to solve that problem, that's as I mentioned at last slide, we need to further identify what the responsible components. So when we find that this could be a single or the combination of some components, to further concentrate, to extract or fraction it together more concentrated, then suddenly you can lower the dose. And another way to do that is that you combine with others in a combination. For example, shrimp oil. We also studied shrimp oil, showed a very nice anti-diabetic effect, and also paper published a couple years ago. And for shrimp oil, you take actually it's just a few grams per day for humans. You can based on the animal trial, you can get effect, significant effect. And of course, other anti-diabetic ingredients. So that's another way is that I said is one is you identify active compound responsible for that, and you concentrate down, and so you can apply in humans, or you combine with other active components, ingredients, and you can lower the dose. And without that, actually, we think about the marketing or commercialization. That's starting with, like, say, fast food market. That could be a niche marketer or a large pet food industry like Nestle. But Nestle, I talked with Nestle pet food research scientists. They are very interested in this kind of ingredients. But they're concerned that is, do you have enough material? Because Nestle, for example, they require a huge amount. Because when I talk with them, I talk about how much we can produce in Canada. But the whole world, like Norway, produce large herrings, and then you make it a whole world, and the supply. And if somebody says, say, one company, okay, I'll collect all of those and process the herring meat and give to a pet company. They can make anti-obesity diabetic food for pets, because that's similar to humans. It's a huge problem in pets. We love animals too much. I guess sometimes give them too much eat and they get obese, diabetes. And they say they have two applications potentially. One is generally used for all pet food. Another is that it reduces the requirement of herring meat. They just put it in a specific anti-metabolic syndrome pet food. That's a kind of significantly reduced amount. And another way, if we still can't get that enough to supply to large companies like Nestle, then you can look at the niche market. There's some small pet food company. That probably we can provide enough. Especially combine Canada and Norway together, then you have a huge amount of milk. And it can start from a pet food market, and gradually it can expand or further develop into a human market, right, for human consumption. That's several ideas that we talk and we think that we want to do more work in the future.
Yeah, but
there are some good and bad news, if I can add, is that we fish the herring in, I would say, in fall for the herring roe, for the female. So we don't do anything with the herring milk. So that's a good product, and it is food grade at the plant. So we really get the nice products for human grade food at the plant. But it's enough if we take only New Brunswick. But if we go and take herring from everywhere, I don't think this is an issue anymore. But I did calculate how much I can get from drying that, and I can get around 200 metric tons of dry product. So it's still not that bad, but those big companies are 1,000 tons needs. So we need to collaborate more. If we could find a market, then we could get it from everywhere. And for us, drying was a good way because we could keep the products for 5 years in a good shape. So you don't have to travel with water. So you want to find a way to share that. And so this is why we are there. But I made like 2-3 more slides. I don't know if you want to share them. Rachel, I don't have them in front of me as a discussion. The 3 slides I sent to you?
Yeah.
Rachel?
Yeah.
I send you the slides, additional slides. Do you have them?
Yeah.
Thank you.
This one?
So just, I don't know if we answer the question of the participants, but I'd be more like that. But what we are really doing here is that we have published, and you will see that paper, that we show similar effect with protein, fish, herring milk protein hydrolysates, and herring milk powder itself. We have developed the technology. We have a picture at the end of the dryer that we have built. And now the problem is really in the dosage. We saw that with Carolin. 4 grams seems not that much, but in tablets, it's a huge amount.
Yeah.
You take 1 grams per tablets, 4 tablets. But if you take it in a functional food, like add a spoon to orange juice or to a milkshake or whatever, it will be possible. But again, don't forget that herring milk is the reproductive organs of the fish. So it's not really so attractive for people to find the active component and make it more concentrate. The issue is to concentration. We have to find a way to concentrate the active products, the active component. And now the second slide, please. So as we saw from that is that we need to, as I mentioned, to find a way, and I guess this is what you did with the 4 grams, you purify, you use the low molecular weights, but still 4 grams is not that enough. You didn't see that much effect. So, and in our case, we know that to get those effects, you need to get more products, more concentrate, and you can do that in animals, but we cannot do that in humans. So we have a challenge that we are facing. So the idea is to use different approach. We use different products. I think the commodity of having product that has combination products like fish oils and some other active products is a good way of implementing that. You don't want to have 3-4 different products, but a combination of a product. So we have to work on that. Obviously, if you add shrimp oils, it's very difficult to do a placebo versus shrimp oil because you see clearly the difference, and I don't know how to mimic shrimp oil's color, the red color of the shrimp oil. So this is an issue that we are facing with human clinical trials, but you did one, so that's very good news. So for us, we think that pet food, as a young one mentioned, is a good way because it can be incorporated in the diets, and they are facing diabetes and obesity in dogs and cats. So again, as mentioned, we think as a supplement, but again, a supplement that is making sense. You don't want to use 50 or 100 grams per day. So that's an issue. That is what so we are looking for collaborators to keep on going on this. And I have 2 more slides, I believe, because I want to make connection with my contacts at Valorise, if I may. Do you have the picture of my colleagues? Yeah. So I am one of the researchers, but we do have a website. It's called Valorise, www.valorise.ca.
Just check all the information. Don't worry, all the details will be available.
Perfect. Yeah. So it's not an issue. Maybe we can finish with the picture of our dryer.
I only have that.
Oh, okay. It's because it was hide. I'm sorry. We forget about that. It's okay.
But thanks. Actually, we have another question from also the attendees. Do you have an idea of the impact of complementing with fish hydrolysate on microbiota?
I saw a paper on that, I guess, from the group of that doing microbiota, but in our case, we didn't do anything.
And you, Carolin?
So we, as part of the project, we were 2 PhD candidates, and my colleague, she did a study in individuals with irritable bowel syndrome, but we didn't, and they collected fecal samples, but we didn't do analysis of the microbiota. We just looked at the symptoms and if the inflammatory markers changed, but we couldn't see anything. But it would be interesting to look at the microbiota.
Yeah, because now there is more and more focus about this topic, and we know that the microbiota has a really important effect on obesity and also diabetes. So, yeah.
Yes.
So, but we are running out of time. So I would like to thank all of our speakers. Thank you for this amazing presentation. All the presentation will be available on our website. The details also from Valorise and also from Carolin Jensen will be also available. Thank you all to attend to our webinar, and see you our next webinar. Bye, everyone.
Thank you very much.
Hey, thank you.
Bye-bye.
Bye.